Do you know, we humans are actually acting as an intruder in their world. Surprised? No, it is the fact. Actually, there are large number of microbes that is in and around our body. Okay. Fortunately, we have a very strong immune system that we are capable of tackling them. But sometimes they prevail and corona happens. Right? This is Dr. Pankaj Kumar, your mentor for today's sessions, and in today's sessions we will be talking about the bacteria, in which we will be talking the introductory part of bacteria, their structure, their flagella, their glycocalyx, right? The kingdom Monera includes the two groups. One is the eubacteria and second is what we call the archaebacteria. The very basis for the recognition of these two groups are the nature of the cell wall, right? Now, you know, eubacteria is often called as a true bacteria. Okay. Can you tell me why? Why we call them eubacteria as a true bacteria? Well, the reason lies in the cell wall. All those bacteria in which the cell wall is made up of peptidoglycan is called as a true bacteria. Okay, and there is only one group of bacteria in which the cell wall is actually not made up of peptidoglycan, and that is archaebacteria. Otherwise, all bacteria are true bacteria. Okay, so once again I would like to repeat, the criteria to define a true bacteria is the very presence of the cell wall made up of peptidoglycan. Okay. Now, the moment we talk about the bacteria, actually it is enclosed by a tough cell wall, right? And cell wall is rigid, and that you know very well, and that is chemically made up of peptidoglycan. Some bacteria are photosynthetic, right? And they have a bacterial chlorophyll in them. Mind it, that photosynthetic bacteria are anaerobic. That means oxygen is not going to be released over there. Okay. And one more important thing is that they do not have a chlorophyll A. That is another important thing, because you see, chlorophyll A is a universal pigment that is going to found in every plant. Okay, but in case of bacteria, we don't find other chlorophyll A, rather it is only the bacterial chlorophyll. Now, bacterial size is important, you know, the bacterial size is just 0.2 to 1.5 millimeter in diameter and 2 to 10 millimeter in length. Okay, so there are some important information like the smallest bacterium, the largest bacterium, and the longest bacterium. Okay, so Bacillus burstyi is the longest bacterium. Bigueotia mirabilis is the largest bacterium, and Dalister pneumocyntes is the smallest bacterium, right? Though these questions are seldom asked in competitive examinations, right? Because you see, in competitive examinations always the conceptual questions are asked, right? Now let's talk about the shape of the bacteria. You know, primarily there are three shapes. Okay, what we call cocci, rod shaped, and spirilla. Okay, so some of these species are pleomorphic. That means that they are going to be present in more than one form, right? For instance, some of the species of rhizobium, so they are pleomorphic, that exist in more than one forms, but basically they exist in these three forms only. Now, what happens that other forms are actually the extension of these forms only. For instance, take an example of Diplococcus. So here you see, Diplococcus is nothing but two coccus or two cocci are actually combining together. Similarly, Staphylococcus, that means the bunch of the coccus cells is coming together, what we call a Staphylococcus, not Streptococcus. Now, if the cells are arranged in a linear fashion, as you can see in the diagram, right, then we call it as a Streptococcus. Okay. Now let's talk about the structure of the bacteria. You know, the structure of bacteria is typically consists of a cell covering by the cell wall, followed by the plasma membrane, right? And within the plasma membrane, what we see, there is a protoplasm, and within the protoplasm, what we see, the genetic material and other things. Okay. So the entire structure we can discuss under three heads. For instance, the surface appendages. So these includes the flagella, the fimbri or the pili, The second, we can say the cell envelope, which includes the glycocalyx, the cell wall, the cell membrane, right? And finally, the protoplasm, where we see the nucleoplasm as well as the genetic material. So one by one, we will discuss about all these things, right? So in this session, we will be talking about the general structure as well as the glycocalyx, right? So first of all, let's talk about the flagella. What exactly is a flagella? Now you see, bacterial flagella consist of three parts. What are they? They are filament, hook, and basal body. So these are the three things, okay? Now what happens? Filament is a hollow body, right? So they are not rigid, they are a hollow body, and they are extension of the plasma membrane. And actually it grows at the peak, not at the base, right? So chemically it is made up of a protein called flagellin, okay? And the basal body is connected to the plasma membrane. One more important thing about bacterial flagella is that they possesses a structure what we call 9+0, okay? And what is 9+0? If I draw a cross section of this flagella like this, then what we will get? We will get that there is a 9 duplet structure like this, okay? And there is no central structure. That's why we are calling them 9+0 structure. Mind it, in case of eukaryotic flagella, what we see, they have a 9+2 structure. So that is going to form in case of eukaryote, okay? It is also time to learn that how we can differentiate between prokaryotic and eukaryotic flagella. That is often asked in competitive examinations, okay? So have a look at this. In bacterial flagella, it is divided into filament, hook, and basal body, right? In eukaryotic flagella, what happens? They have a filament and the basal body. This is the only thing what we find in case of the eukaryotic flagella, okay? Secondly, if you talk about the eukaryote, it is not a hollow structure, rather it is a solid structure. Solid structure in case of the eukaryotes, okay? And in bacteria, it is made up of flagellin, but in case of eukaryote, actually it is made out of tubulin. It is made up of tubulin protein, right? Secondly, in bacteria, it grows at peak, but in case of eukaryote, it grows at base. So this is how we can differentiate between bacterial and the eukaryotic flagella, right? Now let's talk about the types of flagella. Now these are the types. When we say peritrichous, that means flagella is going to present all around the body. When we say atrichous, that means there is no flagella. When we say monotrichous, that is one flagella at one end, right? If I say lophotrichous, that means a group of flagella at one particular end, as you can see in the diagram. Amphitrichous, there are two flagella at the two opposite ends. This is what we call amphitrichous. So depending upon the placement of the flagella, the different types of flagella has been recognized, okay? Now let's talk about another locomotory organ or another surface appendage, what we call pilus and fimbriae. They are mostly found in gram-negative bacteria. So the pili are what we see, the elongated tubular structure that is made up of a protein called pilin, okay? And it is reported in negative bacteria. You can see in the diagram also, right? And mostly the very function of the pilus or pili is during the conjugation, when they form a conjugation tube. They play a very significant role in the formation of conjugation tube during the bacterial conjugation, okay? In addition to that, they have another bristle-like small structure, and what we call them fimbriae, okay? And the very role of the fimbriae is that they helps in attaching the bacteria to any of the surface. That is the very role of pilus and fimbriae, right? Now let's talk about the glycocalyx. That is the outer covering of the bacteria, okay? Now you see, glycocalyx, the very first things, it may or may not be present, right? It is not necessary that all bacteria is going to have a glycocalyx. But if the glycocalyx is there, it can be of two types. One what we call slime layer, and second what we call capsule. You know, slime layer is just gel-like substances that is around the bacteria, okay? And if you talk about the capsule is a tough layer. Again, the capsule is of two types, macro capsule and micro capsule. As the name suggests, micro is a smaller, macro is a larger one, okay? And the size of the capsule, if you talk about, then it is actually in the range of 0.002 micrometer. That is the size of a capsules, okay? Now, slime layer, what is the function of slime layer? They help in attaching the bacteria to the any of the surface. That is the very role of slime layer. But at the same time, it also helps in just providing the some sort of moisture or prevents the desiccation of the bacteria. So these are some of the roles of slime layer. There is a one question. Do you know how our mouthwash works? Because you know, we use mouthwash to just get rid of the bad smell of our mouth. Okay, and what is the reason behind the bad smell of our mouth? The reason being one of the disease what we call pyorrhea. Okay, so pyorrhea is a bacterial disease. What happens? Here bacteria attaches to our gums, okay, and since they are attaches to our gums, right, the foul smell is actually coming out of them. What happens? Mouthwash consists of the hydrolytic enzymes. So when we take mouthwash in our mouth, what happens? This hydrolytic enzyme hydrolyzes the polysaccharides. That is the cell wall, that is the glycocalyx, because we know glycocalyx is chemically made up of polysaccharides. When they hydrolyzes, what happens? The entire bacteria actually get removed from the gum, okay, and immediately the bad smell goes, right? This is how the mouthwash works. I hope the very little introductory part what we have discussed in this session is absolutely clear at your end, right? Thank you.